All right. And with that, we can go ahead and get started. So thank you everybody for joining us on Vincere's weekly journal club, which we have recently opened up to the public. It's a new experiment. We've liked it so far, and we're big fans of open science. So this is great. Thank you for joining us. And we're also joined today by Dr. Laurie Sanders, who is Associate Professor of Neurology and Pathology at Duke University. And she has invented a blood-based biomarker assay that has relevance for both Parkinson's disease and mitochondrial dysfunction, which are our two absolute favorite topics here at Vincere. And our interest, particularly in this, is a very translational one. There are two types of biomarkers that are essential for therapeutic development: target engagement biomarkers and disease progression markers. And this again falls at the intersection of the two, which can be very meaningful for developing therapeutics, particularly mitochondrial therapeutics. So quick housekeeping items. Again, put any questions in the Q&A section. I apologize, the chat is disabled. But if you put them in the Q&A section, we will answer them at the end. You can also raise your digital hand to ask the question yourself, whether you put it in the chat or not. And if you are not on the Vincere team and at any point would like to unsubscribe from these calls, please message Anahit at, and I have misspelled her email in the chat. I will correct that, Campbell, like the soup, at vincerebio.com. And with that, I will pass that over to Dr. Sanders to tell us about her really interesting new work.
Thank you so much, Spring, and thank you so much for this opportunity to share our work. So in addition to running a translational neuroscience laboratory, the other hat I actually wear is I'm the faculty lead here at the Duke Clinical Research Institute, where I lead and design clinical trials in the neurodegenerative space, including Parkinson's and Alzheimer's disease. And so these are the types of questions that I'm constantly thinking about, of the different types of biomarkers that we really need to integrate into our clinical trials, really to get some of these therapeutics over the finish line. So I just wanted to just briefly, I'm going to go over this really, really fast because this is all published, but to kind of like set the stage for where we were when we were thinking about developing sort of this blood-based biomarker. So what we have found to date, and this is in neurons, mice, rats, human postmortem tissue derived from Parkinson's disease patients. And sort of what we found is that there's this shared phenotype of mitochondrial DNA damage, and this is from genetic types of Parkinson's disease, such as LRRK2, as well as idiopathic Parkinson's disease. Now, we know that the G2019S mutation causes mitochondrial DNA damage because we've done those gene editing experiments where we revert back to the wild type sequence using at the time zinc finger nuclease technology, and our phenotype goes away. We know that the kinase activity is required for this phenotype. We've used now, I think, up to 4 different LRRK2 kinase inhibitors. We're able to reverse the mitochondrial DNA damage. To our surprise, it turns out that we discovered this both in LRRK2 models as well as idiopathic Parkinson's disease models, even in the absence of a mutation. What is sort of exciting to us is that these discoveries were made in models of genetic and idiopathic Parkinson's disease, and that we found that mitochondrial DNA damage actually accumulates specifically in the dopamine neurons, of course, the ones that degenerate in Parkinson's disease. And so there seems to be sort of this brain region specificity for where this mitochondrial dysfunction is, in which we can measure. We started because we were really optimistic that we were finding these changes before there was overt neurodegeneration. We began to look, at least in these models of Parkinson's disease, to see if we could detect peripheral mitochondrial DNA damage in blood and muscle and otherwise. And we could, and it seemed to be sort of a good surrogate where we could test sort of all of these matrices all at once. And so sort of taking all of these together, we thought really supported towards developing and moving into human discovery work. This is probably a slide that you have all seen many, many times before. But of course, by the time that someone's diagnosed, they may have had the disease for potentially decades, right? And so there's clearly a need for in being able to predict who's going to go on to have Parkinson's disease, and perhaps intervening earlier may give us a better therapeutic window. But then there's certainly this heterogeneity post that diagnosis in terms of one's journey and in terms of those clinical symptoms and the different motor and non-motor complications that one can experience. And so this is incredibly difficult to model, as well as really be able to harness all of this collectively to understand that heterogeneity that we know occurs in Parkinson's disease. So really, I think some important salient points that have not been considered in previous clinical trials have been that we haven't really considered the complexity of individual differences that might be underlying these pathogenic mechanisms, as well as the possibility that not all patients will display molecular dysfunction in the same biological pathways. And so sort of taking this in mind, we really have focused on mitochondrial dysfunction and its predominance, I think, is in the etiology of Parkinson's disease, and whether we could take sort of our preclinical findings and translate them into developing a blood test for Parkinson's disease. So in order to do this, the technology didn't really exist for us to be able to measure mitochondrial genome integrity. So we developed this assay. It's called Mito DNADX. The principle behind the method is that when the DNA polymerase encounters DNA damage or a lesion, it's blocked and it's not able to replicate past that lesion. And so if you have a template that has little to no damage, you're going to get lots of PCR amplification, like you would in a normal PCR reaction. However, if that template is damaged, such as we know in Parkinson's disease, that polymerase is going to be blocked. You're going to get less amplification. And based on a set of mathematical equations that we developed in the Poisson equation, we can actually then calculate that lesion frequency. So the kind of data that that sort of spits out from Mito DNADX is that, let's say this is sort of your healthy control here, that the more damage that's in that cell is just going to shift the curve to the right. And that's where we're able to calculate sort of that difference. This is a sort of, we're capturing a wide net of lesions that are really going to block that polymerase. So it's going to be oxidative DNA modifications, a basic sites, which is a loss of a base on the DNA, and single double-stranded breaks. We, as of now, are not able to resolve what kind of damage is. We're sort of capturing all of that. Of course, we know that there's all our cells will have different copies of different numbers of copies of mitochondrial DNA and mitochondria, and so we normalize all of our results to mitochondrial DNA. Incidentally, though, so far we've really seen very little differences in mitochondrial DNA copy number, at least in these experiments. So sort of as proof of principal experiment, sort of validating our assay, we treated HEC293 cells with increasing concentrations of hydrogen peroxide, which we know is a sort of potent inducer of DNA damage. And sort of now just showing the data is that sort of our vehicle control here is in the open circles. And then as we treat with increasing concentrations of hydrogen peroxide, and this was only for an hour, you can see that the curves start to shift to the right, indicating that there's more mitochondrial DNA damage. Here it's quantified. You can see that obviously this is causing mitochondrial DNA damage. But importantly, that this is specific PCR product. We don't have lots of degradated products, and we're measuring only one specific PCR product here. That's actually a pretty large PCR product, and so it took a while to sort of optimize that. Here in D, as I said, we always normalize all of our data by copy number, and under these conditions, we don't really see any differences in copy number, but we do normalize that data. We don't really see any changes here, but also that there's This is a specific product for the internal mitochondrial DNA control. Again, there's no spurious products. This is specific. And then when we normalize the data to both 10 kb and mitochondrial DNA copy number, that we see a lovely dose response curve in terms of increase in mitochondrial DNA damage. Importantly, the input has to be high quality DNA that goes in, because really we're measuring those differences, right, in terms of the intactness of the DNA. And so here I'm showing you the input for this experiment, and so it's really of high molecular weight. There's not nicks and breaks and et cetera of the DNA, and it wasn't it was isolated under very gentle conditions, we'll speak to in a minute, and so we're not adding artificial oxidation during the process. So to be able to make this work, we were really thinking about translation. So not only, is our Mito DNADX in a 96-well platform, it can be done in any sort of RT-PCR machine. That's a commonly used equipment in most labs. We also wanted to make the extraction process as easy as possible. And so we optimized with the Autogen 610L machine. It's semi-automated. We can isolate 6 samples in 12 minutes. And our goal was to be able to isolate high DNA template quality, to be able to minimize those pre-analytical variables and enhance throughput. And actually, Autogen actually uses our methods that we developed actually for blood to be able to share with others. And of course, happy to share all of these protocols. You're saying, well, why wouldn't you just use sort of conventional assays, right, to measure mitochondria? Why did you go through all of this to be able to do it? Well, it turns out and we went through this sort of really lengthy process to recover high purity mitochondria. And what we found was that, and of course, these are showing, that we enriched for our mitochondrial DNA compared to, GAPDH. It's almost an 800-fold enrichment from our assays. And we've written extensively about this. I've sort of put our article here that we published in 2018, sort of really comparing mitochondrial and nuclear DNA damage types of assays. It turns out that they're not sensitive enough. So if we use sort of an abasic ELISA or an adeoxyguanine ELISA to measure under hydrogen peroxide conditions, using this enriched mitochondrial DNA, we can't actually detect any differences. And for the adeoxyguanine ELISA, it's actually below the detection limit. And so these just really aren't useful. They're great for the nuclear genome, but they cannot apply to the mitochondrial genome. And so it's just sort of comparing now and just sort of being very transparent about what are the advantages and the drawbacks of this method, is that it's really now a rapid isolation from DNA to where we can get a lesion frequency output. We're able to do this if we start in the beginning of the day, we can get the answer the next day, so that we can get an answer in 24 hours now. Without going sort of into the technical details, we don't need to sort of do cycle optimization because we've chosen to do sort of a nonlinear curve fitting approach. We're actually capturing all of the data at once, and so we can do the experiment in one shot instead of multiple PCRs. We've converted this from a real-time assay, from an endpoint to a real-time assay, and so that really gives the advantage of being able to, you can literally watch it from your phone on an app, the data, and then be able to calculate it very quickly. We've done this in a 96-well platform, but there's no reason why it couldn't be through a 384 and higher. It's just we're really going towards being able to get that higher throughput. What are the caveats? We're not able to identify the molecular types of mitochondrial DNA damage, although we're working on being able to do that, and hopefully be able to share that data soon. And all of our data is relative, which means that we're comparing back to sort of a control, and in this case is healthy controls from humans. So we first started, we knew that in lymphoblast cell lines that there was an increase in mitochondrial DNA damage and LRRK2 G2019S carriers that had a diagnosis of Parkinson's disease. And this was a collaboration with Sabine Hilfiger, where she sent us blinded samples of LCLs, and we did obviously all of the analysis blinded, and then sent her back the data, and this is what we found, is that we also saw an increase in mitochondrial DNA damage in many of the idiopathic Parkinson's disease LCL derived patients. To our surprise, when we treated the idiopathic Parkinson's disease derived LCLs, we saw a reduction back to healthy control levels of mitochondrial DNA damage. And so even in the absence of LRT2 mutation, we're able to rescue the mitochondrial dysfunction with LRT2 kinase inhibitor, very similar to our studies in G2019S Parkinson's patients. Also perhaps surprising, and in neurons, well, this part's maybe not surprising, that when we treat dopaminergic neurons or midbrain neurons with rotenone, we see an increase in mitochondrial DNA damage. But we also treated with a LRT2 kinase inhibitor, we were able to rescue this effect. And so, of course, this is the absence of a mutation, suggesting that that the mitochondrial DNA damage is LRT2 kinase dependent. Really think that LRT2 is sitting perhaps at the sort of a lever almost of mitochondrial genome homeostasis. And the reason I say that is that we found that in transgenic LRT2 G2019S knock-in mouse models in which we isolated the ventral midbrain, and these were studies of samples that were sent to us by Heather Melrose, as well as samples that were derived in my laboratory. When we combine the data, we see that there's an increase in mitochondrial DNA damage, specifically in the ventral midbrain of these animals. I'm not showing it here, but it was also specific to the midbrain and not other brain regions, such as the cortex. But when we do sort of the opposite, when we knock out LRT2, we actually see that reduction in mitochondrial DNA damage. And I'd love to talk about this because while you say, well, that's great, right? We got rid of perhaps some of the damaged mitochondria, but I think what this is suggesting that mitochondria, that LRT2 may be sitting sort of at that role in terms of mitophagy. This is consistent with many publications in the literature. But you also don't want to get rid of healthy mitochondria. And so I think it's going to be really important to be able to maintain that mitochondrial genome homeostasis. So taking these together, decided to sort of now look in PBMCs from idiopathic Parkinson's patients. These first experiments were able to be done because reached out to collaborator Fabio Blandini in Italy. He had a whole bunch of PBMCs that had been in his freezer for decades, actually. Again, we did these studies blinded, but what we found was that in idiopathic Parkinson's disease, in PBMCs, there was this increase in mitochondrial DNA damage, and showing the sort of AUC curve here. But we know that these had been in the freezer for quite a while. We know that there's degradation, the DNAs become sheared when it's sort of frozen as a cell pellet. So we have done a lot of work in terms of trying to mitigate these pre-analytical factors. We showed that if you extract the DNA immediately versus just have the cell pellet sit in the freezer for 2 months, that is sort of trending towards actually increasing mitochondrial DNA damage within the same individual. And even copy number decreases with time in the freezer. And so I'm not showing you here, it was like 4 years of trying to figure all these out. When we sort of once we were able to optimize these pre-analytical factors, I'm showing you now here a prospective study where we recruited idiopathic Parkinson's patients in healthy control. Hopefully what you can appreciate is that we certainly saw the same, I think, effect in terms of an increase in mitochondrial DNA damage, but our range of lesion detection completely expanded. Here we're almost getting to 3 lesions kb, and really we know that we start to see cell death actually at starting to between 1 and 1.5 lesions per kb, and so this is a really high level of mitochondrial DNA damage. But incidentally, the sort of distribution, though, perhaps, is that there is an increase, right? But it's not going to be everybody, and there's certainly ones that have really high levels of mitochondrial DNA damage. We wanted to make sure that it wasn't the Parkinson's disease medicines that were causing this effect. And so really grateful to the patients that agreed to this study. So what we did was that there was an acute washout. Patients withheld their medicine from the night before. They came into the clinic. We took the blood, did some clinical measures, and then they took their medicine, and that can be really uncomfortable. You know, the data showed exactly the same thing, and so we see this quite heterogeneous response in terms of phenotypes. That certainly there's some overlapping with healthy controls, and some that have really high levels of mitochondrial DNA damage. But overall, that there's a higher increase, suggesting that at least in an acute situation that the Parkinson's medicines are not responsible for what we're seeing in terms of our phenotype. But of course, we haven't done this in drug naive patients, and so we would really need to do that to completely rule it out. So we went on to, you know, based on all these findings, we collaborated with the Michael J. Fox Foundation for the FBN cohort, in which we collaborated with 4 academic medical institutions across the country, in which they recruited this validation cohort for idiopathic Parkinson's disease, as well as LRRK2 G2019S carriers that had a Parkinson's disease diagnosis, as well as non-manifesting carriers, so that they carried the mutation but did not have a clinical diagnosis. So those blood samples were collected across these institutions. They were then sent to Indiana University to their biobank there. They were coded, sent to us blinded. We did the analysis and then sent the data back. And what we found was that now in a much larger sample size, that there's quite an increase in mitochondrial DNA damage. We're really excited that all repeated. And certainly, again, has a sort of distribution, right, of differing levels, I should say. To our surprise, if we didn't know the genotype of these individuals, we would not be able to discriminate between idiopathic Parkinson's disease and G2019S carriers that had a Parkinson's diagnosis. These are not statistically different. Perhaps really excitingly is that those non-manifesting carriers, there certainly was this group that had an increase in mitochondrial DNA damage and those that didn't. And so while, yes, I mean, we're really excited that certainly we're in that diagnostic range. I think there's a lot of utility here. But I just want to sort of point out things that like I'm really excited about. And one of them is perhaps your eye is drawn, right, to this sort of 1.5 lesions per 10 kb. So we've done this mathematically where we can completely discriminate between Parkinson's disease and healthy controls. And so we do have that discriminating factor. And we think, at least I would hypothesize, that perhaps this group, at least molecularly, is very different than this group that's overlapping with healthy controls. And so what we're really interested in testing is whether this group may respond perhaps better to a mitochondrial therapeutic, for example. I think just as interestingly is that why we know that the G2019S mutation causes mitochondrial DNA damage, and yet in these patients, they're overlapping with controls, which means that there's some sort of resiliency factors there. So I'm actually even just as equally interested in those that don't have it. And this could be used as sort of a patient stratification tool. I just want to just show that we had robustness across individual academic sites. And so when we break this down by our four different sites, that the answers in terms of the increases across all of our patient cohorts was identical. I won't spend a lot of time on this. There's a lot of data, but we did, collaborated with Dario Lessi's lab, and we did extensive analysis on phospho-Rab10 and phospho-Alert2 935. And those are great, I think, pharmacodynamic biomarkers in terms of that we know that they're responding to MLI2 in these experiments, but they certainly aren't able to discriminate between idiopathic Parkinson's disease and healthy controls or any of the other conditions. So they certainly have utility, but they're telling us, I think, very, very different things. This is not a broad-based neurodegenerative biomarker like NFL, for example. So we looked in Alzheimer's disease in two different cohorts. A and B is from Alzheimer's disease patients that were recruited here through Duke in our memory disorders clinic. And C and D is from a banked cohort that was part of the Pittsburgh Biobank. And in both cases, we saw absolutely no difference between AD and the age-matched healthy controls. And you can see that none of them actually reach over that 1.5. And so to me, this is at least suggesting that there's at least some specificity for Parkinson's disease and maybe Parkinsonism. I want to just share some of the, sort of robustness of the assay and the tools that we have. So actually, a lot of this data was generated right after my lab shut down in 2020. You can see that these are the black circles right here. What's amazing is that we took the same samples that were in the freezer at DNA, which is incredibly stable. And this is the data in December of 2022, and it's almost identical. And so we know if the DNA is stored properly and is extracted within sort of a month time of that cell pellet, that we could reproduce the data at least 2 years later. And we'll keep doing this year after year. We've also shown retest reliability within an individual stable over time. And so if we go, take healthy controls over a 12-week period of time, if we look at lesion frequency or copy number, that it's exceedingly stable over this time and has very little subject variability. And so we believe that the changes that we're seeing in Parkinson's disease are not due to these pre-analytical features, and that it's really a mitochondrial dyshomeostasis that we are measuring. And so sort of just in summary, we think we've identified mitochondrial DNA damage as a shared blood-based biomarker of early Parkinson's disease, idiopathic and Alert2 Parkinson's disease. This may help us begin to understand that heterogeneity, perhaps, in the underlying cause. Since we were able to detect mitochondrial DNA damage in some of those non-manifesting carriers, perhaps this suggests that we might be able to identify high-risk individuals that are in the prodromal stage, and this would extend our window of therapeutic opportunity. As this group knows, it is extremely hard to measure mitochondrial impairment and from live samples and be able to develop biomarkers. But we think that we have now a biomarker that's exquisitely sensitive to mitochondrial impairment and may be utilized in a way to help guide mitochondrial enhancement therapies and other forms of precision medicine. So I'll stop there.
Wonderful. Thank you for that great presentation. I think everybody's, there are lots of questions in the chat, not surprisingly. But maybe I can ask the obligatory question before we get into everybody else's questions, which is, have you looked at CSF?
Yeah, so, when we started this a long time ago, right, the same discussions like everybody else, right, is where you start. And I was of the opinion, I think I still am, is that in order for this to really work and translate, we need to do this in the blood. We're going to need, right? It's not that we don't need to verify CNS engagement, but to deploy it and to get enough people to have buy-in, we're going to need to do this in the blood. And so I thought, if we can't get this to work in the blood, then it's game over. And now we can go the other way and say, you know what, can we do this in the CSF? So that's on the to-do list, but that's why we sort of started the perhaps opposite way.
Yeah, I think that makes a lot of sense. I mean, CSF is very invasive, and even for, in the animal models, it's not easy to collect. So I think you've started the right way, but it still would be nice to
absolutely
know. Yeah, and it's nice that you have the brain, so there is some CNS correlation. But
yeah.
Okay, so everybody else, if you want to ask your own question, please raise your digital hand and I'll unmute you. Otherwise, I will just go down the list and ask the questions here. So first question was from Andy, asking if these specific mtDNA lesions implicate loss of function of some of the mitochondrial encoded proteins.
So as you know, there's very few mitochondrial encoded proteins, right? So our best guess at this point is actually the dysfunction is coming from a nuclear encoded mitochondrial protein that gets imported.
We have our favorite ones. I think.
Yep.
Guesses.
Yeah. So right now they're guesses, but I think it's probably likely nuclear encoded.
Okay, just going down the list. I'm going to prioritize people who raise their hands. So Rick Margolin has a question.
Okay, I'm too excited not to do that. I guess I don't get to be seen, but hello. Gosh, wonderful, fascinating. A couple of quick questions. Have you looked at atypical Parkinsonism, such as MSA, for example, or PSP?
So I have literally just submitted a grant to do just that, right? Because of our Alzheimer's disease results, actually, which were surprising to me, right? Because there's hypoglucose metabolism, there's all these other mitochondrial features, right? But it's not hitting the DNA in the same way. So our next thoughts are, is this a marker of Parkinsonisms? Or can this also extend to maybe ALS or some other, Huntington's disease and other, so that's part of where we're going next.
Yeah. Okay. Yeah. Spring, shut me off. I'll just ask a few more, and then maybe you've got lots of other people who want to. Does this progress? Does the level increase with the progression of PD?
So I can tell you from our animal studies, is that at least in blood, it seems to be pretty flat. So it goes up and then it sort of stays. I don't think that's at all mimicking what might be happening in humans, right? Which is a lot more complicated. We really need to do those progression studies, right? Because we need to know, is this something that just happens early and then it sort of stays, or actually does it go down or up with progression? So that's also at the top of our list to do, which is going to be important for clinical trials, right? We've got to know that answer.
And then just one more. Other genetically driven causes of PD besides LRRK2, pink Parkin, GBA, et cetera?
Yeah, at the time, we got interested in LRRK2 because it happens to be we were collaborating with Ulla Isaacson and Birgit Schula, and they sent these stem cells, and actually LRRK2 was our hit at the time, and that's why we, when we started those studies, that LRRK2 wasn't really connected to the mitochondria, and so it's really funny now looking back that that's where we started, right? It didn't start with the obvious pink1 and Parkin. Those studies are ongoing actually for Parkin and pink1. We just recently published actually that synuclein will drive mitochondrial DNA damage increase, and that you need a 75% reduction in synuclein levels to rescue that. So that gives us an idea of how much synuclein you sort of need in order to drive this. And we're looking at VPS 35 and these others right now to see sort of what the broad applicability is.
Okay, Spring, I'll restrain myself.
Thank you, Richard.
You actually had two of the same questions that I had, so I crossed those off. Tom Wessel, you have your hand raised. Oh, I'm sorry. I need to give you.
How do I lower my hand? Lower my hand.
Hi there. Can you hear me?
Yep.
Yes.
Yeah. Well, I'm thinking along the same lines as Rick. This is very exciting. Obviously, it would be good to know how the data would be stable longitudinally in a given patient population. And what went through my mind, and I think we are all sort of thinking the same way, is that if you have a certain threshold of, say, 1.5 per thousand, would that then dictate a quicker clinical progression? Maybe you'd want to enrich those patients in your clinical trial to get your readout more quickly.
That's exactly sort of what we're thinking, right? Now, of course, this is a hypothesis, right? But if we enriched over that 1.5, are they different clinically over time? And depending on what your therapeutic is, right, would that enrich for what you're testing against? And that's exactly some of our thoughts of how to use it.
Thanks, Tom. Maybe I'll ask a kind of selfish question, and one there's probably not an answer to yet, but I'm curious to hear your hypothesis. So I think the G2019S knock-in mice that you used, Heather's, were the same ones I worked on a lifetime ago. And what's interesting is LRRK2 expression is not particularly high in the dopaminergic neurons in the midbrain. So it's really fascinating that you're seeing the difference in the midbrain and not in the cortex, for example. Any thoughts behind what the mechanism might be by which LRRK2 is having this impact and why we might be seeing it in these neurons specifically?
Right, because at face value, like face validity, right, it's a knock-in, right? So the mutation is everywhere. So why can we detect it right in the midbrain and not in other brain regions? And it didn't show the data in the paper, but we can see in the blood too, right? So there's brain region specificity and cell type specificity. But what's even weirder, right, is that what we've shown is that if you use rotenone, which we know is a complex 1 inhibitor, it gets into cells equally, we've shown that causes mitochondrial DNA damage in midbrain neurons and not cortical. So there is something inherently different in terms of the response to the insult. And I think that's part of the secret sauce, if you will, as to why we can detect those phenotypes and not. And we're trying to dissect that because that's, I think, really going to get us at that vulnerability.
And have you looked at locus coeruleus neurons, for example?
We have not yet.
Okay.
No.
Other like susceptible neuron types.
Yeah,
it's very interesting.
Yeah.
I'll stop asking my selfish questions and go back. There are so many questions. Okay. Jody asks, for IPD patients whose PBMCs exhibit the highest number of mtDNA lesions, does this correlate with any clinical parameters?
Trust me, we looked, right? But it's hard, right? Because the clinical measures were taken while they were on drugs, right? So it's actually blunted probably the in terms of their motor severity and some of the other measures that we're taking, and they're early, right? So it's kind of tough to know, but we looked at every which way to see if there was a clinical correlation, and the only thing that fell out was at the age of diagnosis, that there was a very small sort of it could explain sort of that variability that we see. So we don't see anything in terms of any clinical correlates in this regard. We might see that if we look longitudinal, but as of yet, we don't see anything.
Interesting. Jeremy asks, in the brain, this seems to be specific to the midbrain, at least from the regions you've looked at. Is there also some specificity in the peripheral tissues you've looked at? Like, for example, PBMCs you showed, but are there other ones that are maybe not showing as much of an effect?
So, I mean, that's a great question. We haven't looked at neutrophils or some of the other, or even circulating DNA in the plasma. We haven't looked there. We're starting to do those. We started with PBMCs because that's, they're the easiest to collect, and more of a, sort of straightforward. But considering some of the really interesting results from the Alessi lab in terms of the neutrophils and the responses there, it'd be interesting to see if we see the same thing or not, or there's some differences in terms of the immune cells. I mean, we've done so much work on the lymphoblast cell lines, and so this is at least my gut is at least some of this is being derived from the lymphocytes. That's not necessarily it's at the exclusion of the other immune cells, but I think, I would put my bets on the lymphocytes.
Interesting. So to follow up on a question that Tom asked about patient segmentation, we see there's not this kind of big separation of the controls versus the PD patients. Do you think there will be a line that we can draw for patient segmentation to say, hey, these are the ones with the big mitochondrial deficits? It's not a binary, right? It's more of a spectrum.
Yeah, I mean, I've given lots of thoughts of like what's quantitative and what's binary and where we're going to draw a segmentation, right? And all of those things. I think we're going to be able to do that. It is a quantitative assay. And so I think that, we know let's say LRRK2 kinase inhibitor, right? We drop below that line, right? So it can be altered. I think we could segment probably about that 1.5.
1.5. Okay, looking at some other questions here. Anna, he'd asked about the limits of the sensitivity of the assay. Actually, there are 5 or 6 questions. Let's start with that one.
Yeah, we're in the like 0.84, 0.85 range. We're which of course I think is going to give you mainly the correct diagnosis, but not all the time, right? Can we do better than that? We're working on ways in which we can even enhance our sensitivity more to see if there is sort of a bigger discrimination for that, where we would put that segmentation. That sort of work ongoing. And so while I think it can be used as a diagnostic, I think of it more as a patient stratification biomarker.
Yeah, I'm really excited for that as well. That is one of the major challenges we have in the clinical trials is the heterogeneity of the patients. So
absolutely.
If we can have a more homogeneous patient population.
Absolutely.
Fantastic. Yeah. Another question is actually quite a few. I'm just trying to pick because I don't think we'll have time for.
Thank you everybody for all the questions. I love it.
Could this assay be tried on other tissues? Kayla asks, for example, skeletal, cardiac, or other energetically demanding tissues.
Yeah, that's a great question. So we've done almost every type of tissue by this point. And so we have a method to even muscle, which actually is really difficult. Actually, we have a method to be able to do that. So we can do this assay on any tissue. We can also do this assay on any organism that is of choice. And we've done it now in C. elegans, Drosophila, mouse, rats, humans. It's just the primer, the specificity comes from the primer design. And so we can design it to any of your favorite organism, or cell tissue or whatever.
Fantastic. Younghang asks, says, thank you for your presentation. Do the LRRK2 inhibitors increase or affect mitochondrial function or mitophagy in your hands? And I know Ian Ganley, for example, has shown some data on this. I wonder if you see similar results as well.
So my graduate student, Tara, would be very, very excited that you asked that question because preliminarily, at least in the window where we see changes with our LRRK2 kinase inhibitors, we do not see any effects on mitophagy.
Interesting.
So we think that, again, at least a hypothesis is that whatever mechanism that we're is underlying this. And there's another graduate student, Ivana, would be very excited that you're asking this because she's working on that, what we think that black box mechanism is. There's an initial mechanism that is sort of either for the damage or the repair of some sorts. And then mitophagy comes into the picture. And so we think it's a secondary mechanism, but there's another one that precedes it.
Do you think it's a black box? I know there's no answer, but I think the LRRK2 field is quite enthusiastic about the lysosome side of things, which of course mitophagy involves the lysosomes. There's a lot of communication back and forth there, but are you looking at the lysosomes at all in this context?
Absolutely, right? Because they're so you can't disentangle that, right? In terms of lysosome. So I can tell you that at least in terms of our rescue experiments, we don't see the rescue on the lysosomes either in that time frame. So it's not that I think that it's not the lysosomes or mitophagy that's contributing. Absolutely. But I think it's just coming later in the process.
So it's you think it's direct to mitochondria, basically? Yeah. Very interesting. And if I can just add a comment here as well, because I think this is underappreciated. Heather Melrose did show in her paper with the G2019S knock-in mice, and I was in the lab at that time where we had these fragments or what looked like arrested fission on the mitochondria. Yeah. And I think the field was more interested on the other side of it, but it's definitely something that's been shown, I think, by multiple groups now.
Yep, absolutely.
Very, very interesting. Okay, let me go back to the questions here. It's interesting to see how much more variable the mtDNA lesions are in the IPD compared to healthy controls. Do you have any thoughts on what exactly is driving, and maybe this is what's driving that? Maybe this is coming back to the question of mechanism again.
So so at least in my philosophy in my lab, right, is that we do the experiments to kill it fast. Okay, so we recruited anybody that was over the age of 50 and that did not self-declare that they had a neurodegenerative disease. Okay, that's who we recruited to do these studies. And we didn't restrict, there was no exclusion on any other comorbidity or drugs that they were taking, right? And so anybody over the age of 50, is likely, right, or their age range that we're looking at are going to have, they're going to be diabetics, they're going to be all these, right? Because it's that, okay, well, if it's all over the place in healthy controls, it's game over. But it's not. And I think the reason is that there's such evolutionary control for mitochondrial genome homeostasis, because everything follows from that, right? Because your DNA, you start to have DNA instability, you're going to now interfere with replication, transcription, right? Other metabolism, you're going to get cytochrome C, right, release. That has to be maintained to maintain all those other processes. And so I think that, through this experiment, we've seen this now over a decade, right, of that there is a very tight control and there is very minimal variability between healthy subjects and even over time. Because I think when you start to lose it, you're really showing disease at that point, and not something that's just at least in a healthy individual.
Yeah, that is really fascinating, especially that it is in the periphery. It's nice that you showed the Alzheimer's data, because that's, an obvious question about other neurodegenerative disease. Can you differentiate? So this could actually be really good from a diagnostic perspective. Yeah. It can differentiate. Are you thinking about, I know this opens up opportunity to look at so many different things, and I know you have limited time and resources, but what about patients that are kind of at the intersection, like Lewy body dementia, or maybe more of the synucleopathy type diseases that are not Parkinson's?
Right. I'm just intrigued with the overlap in terms of the pathologies across the different neurodegenerative, spectrum, right? And like synuclein and AD and beta amyloid and PD and like all this, right? But clinically, right, they in certain contexts will look very, very different, right?
Yeah.
What we're trying to build here at Duke is actually being able to assess all that are coming through our clinic. And so that we can get a better idea of exactly that if you end up in the memory clinic or you end up in the movement clinic, right? Because that's a whole nother thing, right? No matter where you ended up, clinically we have your diagnosis. What do you look like on our test?
Yeah, it makes sense. And I did just realize that Rick had asked a similar question here. One other question from Kayla regarding the assay itself. Have you done any, she asked, periodic blood draws, and she specifically asked about the rodents, but I also wonder about the patients, how consistent is this within the same patient? I know obviously there's more heterogeneity across different patients, but within the same patient, do you see kind of a similar trend over time, or?
Yeah, that's interesting. We haven't done it actually. So we've done it in the rodents, the blood, and that's pretty stable. We did it like one day, 5 days, and actually it's not in the paper, but we also did it sort of at endpoint, and they all looked sort of the same. In humans, we know at least over a 12-week period within an individual looks similar. We now have data for a year. With the same individuals in both healthy controls and Parkinson's disease. I don't have that data analyzed yet, but we will have that very soon. So we're going to know exactly that answer.
Awesome. You're very efficient at answering the questions, I have to say. I did not, when I saw all these questions coming, I thought there's no way we'll get through them. So on that note, and I know we talked about this just a little bit when I saw you last time, but have you looked at some of the other biomarker biomarkers that are coming up for Parkinson's disease? I think the synuclein seed assay is the one that we talked about, but we've also heard about phosphoubiquitin and potentially Miro as biomarkers. I wonder if you've seen if you looked at any of those, seen any correlations, or is that in the plans?
Yeah, I mean, that's certainly the plans. I mean, I think that each of these biomarkers are really significant and different in what they're telling us about the patient population. Mm-hmm. And that, at least my approach is, I think we have to complex them together and figure out which ones are correlating and which ones aren't. And that 30% or whatever that's SAA negative, is that our 30% that's actually higher? We don't know, right? And I think it's really going to take that collaborative effort to be able to look at these all together to really understand, so that we can define those homogeneous populations to be able to get these therapeutics over the finish line.
Yeah, I agree 100%. And if there's anything we can do on that front to help out, let us know. It was interesting. I'm not sure if you saw there was a presentation at the Fox Therapeutics conference just last week that was talking about Miro, and they mentioned that they see correlation with the synuclein and Miro, which is a bit surprising to me. I would have thought maybe it would be the opposite, maybe. Yeah. Although there is clearly a relationship between alpha-synuclein and mitochondrial function as well. We've seen that now, from several different groups. So maybe it does go that way. But yeah, I'm eager to see what those results will look like.
Right. Yeah. I mean, I'm a little surprised by that. But it's also hard to know, right? Because the synuclein is sort of a yes, no. And so, it's 2/3 of the population, right? And so, and certainly I think mitochondrial dysfunction is probably 2/3 of the population of sort of those markers. So, I'm really eager to see how all these is going to fall out.
Yeah, I agreed. Exciting times for Parkinson's. One technical question from Jeremy. He says, very important work in figure 4, the level of mtDNA lesions in healthy controls was similar to that of the immediately extracted DNA in panel D. And I'm not sure if you remember that, those panels, but the validation group showed almost no lesions, thus significantly improving AUC to 100%. What reagents did you add to the validation PBMCs for storage?
Oh, yeah. So, I think this is statistical probability, right? And it's a relative assay, right? So it's whatever was sort of grouped at that time together to sort of normalize back to. And so everything's kind of set. If you think of it like a Western, you said it's sort of to 100. And so it's going to look similar, but they weren't really similar in terms of absolutes when we were doing it. I think that initial validation cohort where I mean, it is true. That's what we found, that there was no variability with those 6. But I think it's just our N wasn't high enough to see that variability. Like we just literally lucked out with, I was like, oh my God, we have to report that this is 1. I don't think it is 1, but that's like what we found, right? And you can see with the further cohort, it's not 1, it's not perfect. It's just that literally statistical probability that of those 6 individuals, there was no variability.
Interesting. One question from Rick Margolin asking, considering mitochondrial trafficking in neurons, could you look at fractions, for example, synaptosomes, to see if there's differential damage in different parts of the neurons? And then can the assay be used at single cell level, for example, in microfluidic chambers?
Oh, so that's a great question. So we haven't looked at mitochondrial DNA damage, but we have looked at mitochondrial biogenesis in those microfluidic devices. And what we found, certainly, at least in a rotenome model, is that it's certainly happening at the synapses and the processes well before it's happening in the cell body. So that's consistent, at least, with a dieback mechanism, right? That everything's going to be happening at the terminals, the mitochondria become dysfunctional there, and then there's going to be sort of this dieback. We have not done sort of the separation between those two to see like where our mitochondrial DNA damage is occurring in those types of experiments, because we need more material to be able to get enough high quality DNA, because it's not just DNA, we need high quality DNA. And so we are limited in terms of sort of it's not a problem for blood or whatever when you have a lot of material, but for neurons it's a little bit more challenging. So what we haven't overcome is to be able to do those experiments or single cell. It's just not enough material, at least in the iteration that our assay is in.
A follow-up to that too, for the brain that you showed, you looked at midbrain. I'm assuming if you're just collecting midbrain, you're looking at cell body region, right? Have you looked at striatum for axon terminal regions? Maybe, of course, you get the medium spiny neurons there too, but
yeah, so we haven't really seen too much in the striatum.
Interesting.
Yeah, I mean,
again, interesting, because the medium spiny neurons have high expression of LRRK2, so you would think, yeah.
Yeah, so that doesn't seem to be it. We at least know that when we look at postmortem human brains, of course, there's a ton of degeneration, and we're looking at the graveyard, right, of what's left. But at least there's certainly a higher proportion of, it was using a basic histochemical assay that we developed to be able to look at mitochondrial DNA damage like in a slice, that it certainly was in the cell body at that point. Didn't seem to be really in glia, which is fascinating. So it seems to be sort of neuronal. It's hard to say because most of the processes were kind of gone. So we didn't really see a whole lot, but I don't know what's happening sort of in the earlier part of the disease process.
Okay, one last question. We have 1 minute. Have you looked at whether this correlates with age?
Right, so that's also $1 million question, right? Because in the theory of aging, right, free radical of aging, you would suspect, right, that this changes over time. At least in our human studies, what I can say is we do not see it correlate with age. And damage is different from mutations. Mutations may correlate with age, but damage does not. And so we think we're looking at a disease process and not it is age dependent because it's stochastic, but this is not an age-related effect.
Interesting. Do we have time for one more?
Sure, go for it.
All right, we have a question from the audience. I've given you access, and
Okay, thank you very much. My name is Matthew Onikoko. I really appreciate the presenter for that wonderful presentation. Most of my questions have been captured from Dr. Berus there, but I would just like to know, are you looking anytime soon about interracial comparison, say between Caucasians and, for instance, African? I quite appreciate that last question on age differences and other biomarkers, synchronizing them together. But I really want to know, are you looking into, because like you were talking about some shocks and surprises that you saw in the lab while you were trying to do this work, I think you may find something very shocking too if you do interracial comparison. Thank you.
Yeah, thank you for that question. I mean, you're bringing obviously up a very, very important question, not only ethically, but also, of the recent, studies done in Alzheimer's disease and showing certainly differences in biomarkers across, different, ethnicities. So this is really important. We did not have a high enough statistical N to be able to see if there's any sort of differences. I can see at least in our healthy controls, which were not all Caucasians, but there was no differences, at least we can say, but I can't say that with certainty because we didn't have a high enough N. We need to do better in our forthcoming studies, and that is definitely a high priority for us in terms of both, our controls without disease as well as our Parkinson's disease population.
No, thank you very much. Thank you very much. I may try to reach you much later further on this. I don't know if I'm permitted to do so.
Can people reach out to you, Dr. Sanders?
Oh, of course. Yeah, please. It's easy at Duke. It's just laurie.sanders@duke.edu. So please reach out to me anytime. I'd be happy to chat with you further.
All right, and we're 2 minutes over time, so I'm gonna stop us here. We had lots of questions. Thank you so much for leaving time for questions, because that's kind of the fun part of the process. And thank you again so much for presenting. We look forward to seeing all the exciting new things that will be coming out of your lab.
Great. Thank you, everyone.
Bye-bye for now.